The second lecture on adaptation basically will not be so much about criticizing adaptationism, but trying to provide you the tools to identify, to recognize an evolutionary adaptation. And essentially, we can start right into the core of it. There are certain criteria. There are certain criteria to consider a trait as an evolutionary adaptation. The first criteria is the immediate benefit criteria. Essentially, the trait gives the individual an advantage over other individuals that do not have that trait. Of course, there has to be natural variation on that trait. Immediate benefit. The second criterion is that natural selection has to favor the favorable or the good variant of the trait because of the immediate advantage it gives. This is meaning actually that natural selection operates on the trait in question. So, immediate benefit, natural selection operating on the trait. The third one is that this trait has to be inheritable. Because if it's not inheritable, what's the point? The trait has to be inheritable. Once it's been selected for, essentially has to become genetically fixed or assimilated. And finally, and that's the trickiest part, that the adaptation was originally selected. When this trait was selected, it was for the same purpose as it is currently advantageous or beneficial. And for this criterion, we need to have knowledge of the historical origin of the trait. In summary, immediate benefit, positive selection, inheritable, and historically traceable for the same purpose. Let's go through these criteria one by one. Criterion 1, the immediate benefit. The trait confers individuals that have it a benefit. If all individuals have it and benefit from it, can you call it still an adaptation? Well, this is a question of a timing. You can call it an adaptation because nowadays this trait has been fixed. All animals, all individuals show it, but once upon a time there was selection of it. I would say yes, of course you can call it an adaptation. The variation of this trait existed once upon a time. Now it has disappeared because this character has been fixed. Immediate benefit can be studied with common experimental methods that quantify that benefit. Physiological methods work very well. So in physiology, to understand physiological systems, we can see basically what is the benefit for a given trait. Let's look at an example. The example is lamellar recruitment in hypoxic conditions in crucian carp. Crucian carp is a fish. Lamellar recruitment has to do with recruitment of surface area in the gills in terms of exchanging oxygen in the gills. This is actually demonstrated in a paper published in 2003 by the group of Göran Nilsson in Oslo. And what they show is that in crucian carp, this is the gill arch of a crucian carp in normoxic condition. That means in normal oxygen conditions. And then they put these animals in hypoxic condition, that means water with less oxygen, and see what happens at day 1. You start having a bit of a protrusion of the lamella. At 3 days, even more. At 7 days, more. 14 days, full protrusion of the lamella. With recovery, back to normoxic conditions, disappearance. What does this mean? These animals are basically increasing the surface area of the gills in terms of obtaining more oxygen from the water, because the oxygen in the water is low. This would be a classical example of an immediate benefit of lamellar recruitment. When you need it, you put them out. When you don't need it, they are brought back. So I would say that in this case, this trait, the extension or the protrusion of lamella in hypoxic conditions, is immediately beneficial for the animal. Criterion 2, the existence of a positive selection signature. At some point in time, it could be now, or it could be in the past as well, the trait is or was selected upon from other variations in the trait that are or were less favorable. Of course, the focus in this case, it's not on the immediate benefit for the animal, but basically that there is a pressure in the population that is showing natural selection. We have an example. the length of the horns in the horned lizard. And this was a nice paper published in Science a few years ago, how the horned lizard got its horns. And what is interesting about these animals is that these lizards basically have the horns here, the parietal and the squamosal on the sides. And there is a predator on this lizard, shrikes are birds, that capture these lizards, impale them in branches of trees, and eat them from there. Why is this relevant for this paper? Because you leave your carcasses behind. These birds leave a signature of the predated animals. What the authors did simply is go into the field, find as many as they could, sampled animals from the same population, in the same location, animals that were alive, and compare them. And what they could demonstrate, and that's what you see in these survival curves here, is that the animals in the population that live in the population in the blue, here and here, this is for the back horns here, those are the parietal horns, and these are the squamosal horns on the side. What they could demonstrate is basically that in the living population, the horns, both of them, were longer, in this part, longer than the animals that had been predated upon. So what they demonstrate here is that there is a natural selection pressure for having longer horns. Longer horns means higher possibility of survival. So in this case, you have a nice example showing actually that there is a positive selection pressure for actually horn length. Animals that have a bit shorter horns have a higher probability of being predated upon by these birds. Criterion 3, the trait is inheritable. The selection pressure for the trait that we saw in criterion 2 basically caused the genetic assimilation. So that basically this trait now is fixed, and then all the individuals pass it on to the offspring and the next generations. The problem with this concept, and this has been always the Achilles tendon of basically evolution theory, because we do not know how this change occurs. Not even today, we have no understanding of how a trait can be genetically assimilated. Genetic assimilation means that basically some phenotypic plasticity, changes that occur in the body, are eventually make it into the genome and they are inherited. This is the Achilles tendon of evolutionary theory, and we still don't understand the mechanisms. It does not mean that it does not happen, but we don't understand how it happens. The same thing is here. I have to say that criterion 3 basically, criterion 3 usually is assumed in many studies of adaptation. Sometimes you can prove it, but sometimes it's just assumed, because it's really difficult to understand the genetic basis of a trait. That's science in itself, if you want. So you assume rather than measure heredity. But of course, you can think of situations in which genetic assimilation is possible. Genetic drift is the classical example. But of course, it's unlikely that it happens by genetic drift in all cases. But that's just one example. And finally, and this is the most tricky criterion, and that's the one I'm going to spend most time on, is the historical origin of the trait. Because you need to demonstrate that the reason why the trait was selected, when it was selected, it was the same reason that it is beneficial today. Why is this important? This is important because selection pressure operated for a reason. And that reason has to be still relevant. If that's cannot be demonstrated, you could not regard the trait as an adaptation. You should call it a pre-adaptation or an exaptation. It's it's a beneficial trait, but it did not appear for the same purpose in which it's actually used nowadays. And of course, this brings immediately the question how to trace the history of a trait. And the immediate answer is, well, you study the fossil record. If you study the fossil record, you look for ancestral, or you look for older fossils that were related with the current species, and you could prove something. But unfortunately, many characters, many traits do not fossilize. If so, you would be restricted to very, very few traits. There is another method, the comparative phylogenetic method, that I'm going to try to describe to you. because that's actually the most powerful method we have today to demonstrate the historical origin of a trait. And it can be applied to any trait. It does not have to be fossilized at all. The comparative phylogenetic method has as a name to study adaptation indirectly. How do we study adaptation indirectly? Looking at correlation, correlated evolution among characters or between characters and the environment. Where? In the species that are related with each other. The comparative phylogenetic method can be directional. Directional means that you go backwards. You look for the ancestors of the species you have, and then you try to find what are the common or the not so common traits here. Or you can also use non-directional comparisons. Non-directional comparisons use actually phylogenetic relationships between contemporary species. It is possible nowadays, as you know, to take different species that exist and decide or discuss how long ago do you have to go back so that these species were one. If we assume that all species have speciated from an ancestor, we can always take this species, look back and say, well, when did they diversify? In this case, phylogeny helps us identify independent evolutionary events. And these independent evolutionary events are the ones that have a statistical significance. I'll show you what in a second. Let's take an example, and I hope that you will see it with the example. The example is the sperm competition hypothesis in bats. I'm sure you have heard about the sperm competition hypothesis. The sperm competition hypothesis goes about the correlation between group size and the size of the testes. The idea is that animals that live in small groups or isolated, there is less sperm competition. If you have less sperm competition, perhaps you don't have to bother so much about inseminating females with a lot of sperm, and for that you need large testes. But what happens if you live in groups, in large groups? There, the potential for competition between males is much higher. Therefore, which males are going to be fathering the offspring? Hopefully those that can produce more sperm can inseminate more females. So the expectation would be to see a correlation between group size and testicular mass. The larger the testes, the more sperm you can produce, and you would expect that to correlate with group size positively. What do you see in this data set? This data set is for some bat species, and it would seem to support this statement, the sperm competition hypothesis. There is a problem here, however, and that problem is illustrated by, well, I'll show it in the next example. This is explaining what I just told you. Because the prediction would be that larger group size, larger species that live in larger groups offer more opportunities for multiple matings, therefore there is more sperm competition and larger testes. This is an oversimplified theoretical example. Let me start here to explain the basis for it. Now, imagine that you have 6 species that could be 6 species of bats. 6 species of bats that distribute here. They provide you a very good correlation, right? Okay? Group size correlates very well with testicular size. The larger the group size, the larger the testes. If now we take these species and we put them in a phylogenetic tree that looks like this, what is it that we get? What we get is that A, B, and C in this phylogenetic tree basically cluster together. Do you see that? So they cluster together here. For D, E, and F, they cluster together here as well. How relevant is what we are observing here in terms of explaining our sperm competition hypothesis? Although you have 6 species, in reality, you have one single element of variation here, because all these 3 species are related with each other. They are highly related with each other, much more related with each other than this one here. So they come from a common ancestor. These common ancestors here and here at one point may have differed. And then, in this case, the D, E, and F species may have become more social species in larger groups, and the other ones have not. But in this case, what you have is a single appearance of, you could say in this branch, a single appearance of increasing group size with increasing testicular mass. So the strength of the evidence for an adaptation here is very weak, because it has only happened once. You have no statistical evidence. It has only happened once. In reality, you look at this correlation here, but you should be looking at this one, because all these values should be clustered as 1, and all these values should be clustered as 1. Of course, between 2 points, there's always going to be a good correlation. So this is not good enough in terms of demonstrating the sperm competition hypothesis, if that's what you want to show. Now, this is an oversimplified example. Let's look at our bad example. What basically we are talking about is this normalization for phylogenetic relatedness. If 3 species are very related to each other, they are not giving us the same information that if these 3 species were independent from each other. Let's go back to our bad species and all the different points we have here that come from these different species. We put them in a phylogenetic tree, and now we calculate the phylogenetically independent contrasts, taking into account how different species are related with each other. And we come up with this number of points here. Notice that these points now are expressed in a different scale. You don't have the same number of samples here, and they appear in different positions. And that's because we have taken into account the relationship between species. When we take this into account, we still see a good correlation. Therefore, what this is telling us is that now we have support for the sperm competition hypothesis. Because every time in this phylogenetic tree where there was an increase in group size, there was also an increase in testicular mass. I think that this is basically a very simple example trying to illustrate the importance of phylogeny and how related species do not provide the same level of information than non-related species. The comparative phylogenetic method is a powerful method to distinguish cause and effect and eliminate potential confounding variables. Because remember that correlation is not causation. In this case, when you went back here, correlation is not causation. This could correlate, but maybe it's not explaining anything. But once you are able to put the phylogenetic tree in context here, you may be able to explain actually this causation. Phylogenetic trees allow the distinction of direction of causality. Considering phylogenetic relationships numerically allows you to treat the data statistically, which is important in this context to provide how strong your evidence is. Of course, the comparative method can only be applied if the trait appeared independently in different phylogenetic groups. So there is a limitation to the comparative phylogenetic method. You cannot apply it to everything. But when you can apply it, it might give you the proof you were looking after. Let's look at a final example, and with this we'll finish. Is regional endothermy an adaptation in scombrid fish? Regional endothermy is the situation in which in some region of the body, in this case of fishes, they are able to trap heat and keep this body part at a higher temperature than the rest of the body. We know that in scombrids, only a few species have endothermy or are regional endotherms. And this endothermy allows the increase in temperature in selected parts of the body for particular functions. We can have a physiological question here. How do they do it? How do they take that into account? That would belong to a physiology course, but we're not going to bother too much about this. We have tunas that have a counter-current heat exchanger in the muscles. We have billfishes that have a counter-current heat exchanger in the eyes for increasing visual acuity. But that's not what we are talking about in this lecture. What we're talking about in this lecture is the evolutionary question. Why do they do it? The ultimate reason. And this is something that you can answer by now putting these animals, the scombrid fishes, in a phylogenetic tree. And notice what's interesting in this case is that you have the appearance of endothermy in 3 lines in this tree. The red line is the endothermy of red swimming muscle in tunas. You have its appearance here. All other gray lines are actually ectotherms. That means animals that do not show regional endothermy. Notice that in one species here, you have a heater organ in lateral rectus muscle. So basically, this is a heating organ in the muscles in the eye. And then in the green ones, you have another muscle in the eye that actually belongs to the billfishes. What's interesting here is that in the phylogenetic tree of scombrid fishes, you have the appearance on 3 occasions of cases of endothermy. So, of course, when you look at this from a phylogenetic perspective, you can actually get more evidence. Every time an animal has had a particular need, we suppose, to heat up or to maintain a region of the body at a higher temperature, it has come up with a similar solution, regional endothermy, but a different method. It's different muscles in this case. But it's the same solution, or in this case, with the body muscles. So in this case, we could say that endothermy in scombrids is actually a triphyletic trait because it appears 3 times. And it's different every time. You have much more evidence of the relevance of regional endothermy than if all these animals were actually clustered together in the same branch of the tree. And that's how I wanted to finish this. Thank you very much.